Bacteria by Dr. Anne Seignalet, taken from various pages of his book, L'alimentation ou la troisième médecine, which has not been translated. I would translate the title as Nutrition, the Third Medicine. Dr. Saynyalay died in 2002, so this was cutting edge in 2002. This is from the last edition of his book. So, two problems with bacteria, says Dr. Anne Seignalet. Number one, selection by mutation for antibiotic resistance. And by the way, we now know that, in the last 10 years there have been tremendous advances in technology and tremendous strides in looking at bacteria, which I will go into in another video. But we now know that bacteria resistance can be transferred laterally from one bacteria to another and from one species to another. So that's number one, he says, selection by mutation. And B, dysbiosis, which can result in candidiasis. And medicine's narrow focus on infection by complete bacteria is a mistake, he says. We should also be worried about macromolecules, peptides, and these are very abundant. Saynyalay thinks that they penetrate damaged intestinal tight junctions to cause three kinds of pathology, and these are Saynyalay's own classifications. Well, there's autoimmune, classic autoimmune pathologies. He accepts all the diseases which are called autoimmune, he calls them autoimmune, but he actually says that they are xenoimmune, so that it's not the body attacking itself, it's the body attacking these macromolecules, these peptides which have penetrated the gut wall. The other classification is clogging, where the macromolecules clog up the cells and tissues, intracellular spaces and in the cells as well, and affect proper functioning of those tissues, and elimination diseases. And what Saynyalay means by elimination diseases are things like eczema and asthma. And he says these diseases are the result of the body trying to eliminate the macromolecules. Then he says antibiotics disturb bacterial flora. Well, that's well known. That's called dysbiosis. But he says it's also likely that they damage the enterocytes, which are the cells which line the intestines. In the digestive tract, says Saynyalay, there are 10 to the power of 14 bacteria, which I think is 1,000 million bacteria. And he says there's 10 bacteria for every cell. Well, this is quoted in practically every article you will see on the human microbiome. But it appears that the authors are just quoting each other. The latest research, and we'll flash it up, says that it's not 10 bacteria for every human cell. The ratio is actually 1 to 1. Anyway, from birth, says Saynyalay, bacteria from the air and food colonize the gut. And he says these are the quantities. In the mouth, there's a medium amount. In the stomach, there's only a few because gastric acid destroys 99% of germs. In the upper intestine, the bacteria are quite numerous, 10 to the power of 3 per milliliter. That's 10,000 per milliliter, or 10 to the power of 6, which is 10 million per milliliter. So, so it varies quite a lot there. Very numerous in lower parts of the intestine. So that would be the ileum before it gets to the colon. And that's 10 to the power of 6, or 10 million per milliliter, or possibly 10 to the power of 7, which is 100 million per milliliter. And then in the colon, the bacteria are extremely abundant, between 10 to the power of 9 and 10 to the power of 11. So I think that's probably between 1 billion and 1 trillion per milliliter. So that's 10 to the power of 9 is 10,000 million, and 10 to the power of 11, a million. So yeah, million, I think that's 1 trillion. So bacteria form 50% of the weight of your stool, and that's well recognized. Now then he goes on to talk about types of bacteria, and there are only, in the upper part of the digestive tube, only aerobic bacteria. That's in the duodenum and the jejunum. And these aerobic bacteria are successively replaced by anaerobic bacteria as we go down. And just my parenthesis here, lactic acid bacteria are facultative anaerobes, so they can survive in aerobic conditions and anaerobic conditions. And there are quite a lot of bacteria that are facultative anaerobes. So in the ileum, anaerobes predominate and make up 99% of the total in the colon. And he says there are 400 species of bacteria. And this, again, this is outdated. Remember, this was cutting edge in 2002. But since then, there have been fantastic advances in technology for looking at bacteria, all kinds of advances. And it's now known that there are 2,000 species. And there are various, like the human metabiome, there are now, I think, 3 studies on the microbiome. It's now thought that there are 2,000 species. So there are 2 types of flora. There's a transitory flora, which cannot develop further. And there's a resident flora, which can multiply and take up residence. Resident flora is found mainly in the bottom of the intestine and particularly in the colon. The intestinal flora is influenced by several factors, and he gives 7 factors. The first one is gastric acidity. In cases of insufficiency of stomach acid, the bacteria multiply by 10,000 in the upper intestine, with increase in anaerobic types and lactobacilli. So if you're taking proton pump inhibitors and you suppress your stomach acid, the bacteria is going to multiply by 10,000 in the upper intestine. And then number 2, peristalsis, and that's the muscular movement which forces food down your digestive tract. And especially in the upper part of the digestive tract, the germs are pushed downwards in peristalsis. And then there are, number 3, there are interactions between species which can either inhibit or facilitate the growth of other species. And so now we know that bacteria produce bacteriocins which can kill other bacteria, either bacteria of related species or even very different species. And also bacteria can cross-feed. One bacteria that produces one kind of metabolite, another species of bacteria can feed on that metabolite. So they can feed on each other's metabolites and form a colony. and that's when you get a biofilm. Number 4, diet, and regimes which are very rich in meat produce a so-called putrefaction flora, and that's when you get fermentation of proteins. And a vegetarian diet produces a maceration flora. And number 5, antibodies secreted by plasmacytes of the gut wall, and secretory immunoglobulin A in particular, which inhibits proliferation of microbes and prevents them from adhering to the gut wall. Number 6, mucus, and this stops the action of certain bacteria, and on the other hand protects others against the destructive effects of some antibodies and digestive enzymes. Number 7, the capacity of some bacteria to adhere to specific receptors of the epithelial cells. This can alter the structure and functions of the villi. And just another parenthesis, there are some pathogenic bacteria. They attack the tight junctions between the enterocytes and damage the enterocytes. Seignyalay died in 2002, and he laments the lack of interest in gut flora by researchers. And this has changed monumentally in the last 15 years with these new technologies, electron scanning microscopes, and all kinds of things to look at bacteria now. And they're now looking, bacteriocins are the new Klondike. Everyone's looking at bacteriocins as the new, and these are peptides which are produced by bacteria to kill other bacteria. So these are being looked at to replace antibiotics. And I think it's just going to open another Pandora's box. Just going to be another nightmare like antibiotics. 2 classic states of flora, and that's my own opinion, by the way. That's nothing to do with Seignyalay. Bacteriocins were not, I don't think he knew much. I don't think anybody knew much about bacteriocins in 2002. He says there are 2 classic states of flora which are recognized. Saprophytic, and that's where bacteria make a living in the human gut lumen without doing any harm. And there is even a symbiosis where the bacteria are doing beneficial things. They get to feed on products of our digestion, and in return they complete digestion of some foods, break down bile pigments, help to make vitamin K, impede the growth of yeasts and fungus, and release polyamines, which in physiological doses feed the enterocytes. Okay, that's one state of flora, and that's the saprophytic flora. And then there is the second state of flora is the pathological state, where there's an excessive proliferation of dangerous bacteria, and this can cause a disease either by releasing a toxin, and he quotes for the example of E. coli or Staphylococcus, or by damaging or crossing the gut lining, and he gives the example of Shigella or Salmonella. Apart from these brutal accidents, says Seignyalay, he thinks that the intestinal bacteria, and particularly the aerobic bacteria of the upper part of the intestines, are the cause of many other diseases. And Seignyalay discusses Klebsiella as a cause of ankylosing spondylitis, Proteus mirabilis, implicated in rheumatoid arthritis, and Yersinia enterococcolita in Graves' Basedow hyperthyroid, etc. Most bacteria in the intestine are dead, says Seignyalay, and this facilitates their decomposition into peptides, lipopolysaccharides, and other substances which are more or less noxious. So, says Seignyalay, to explore what are the relations between intestinal bacteria and various pathologies would be very useful, and inspiration can be taken from the recent work which has clearly shown the relation between Helicobacter pylori, a bacteria found in the gastric mucus, and duodenal ulcer and some stomach ulcers. The right colon is the first part of the colon, which goes from the ileocal valve to the right colic flexure, and the right colon, and it's on the left if you're looking at a diagram of it. That's my parenthesis there. It contains a bacterial flora which is abundant and varied, mostly anaerobic bacteria, but some debris of aerobic bacteria, particularly peptides, can reach the colon. It's been proved that the wall of the colon absorbs some substances which come from bacteria metabolisms. Yes, these are called short-chain fatty acids. I think in 2002, maybe, not much known about those, produced by the fermentation of insoluble fiber by bacteria, by anaerobic bacteria. So, yes, so see my videos on sauerkraut, and particularly the medicinal effects of sauerkraut, and my protocol for C. difficile. So that's it.